main function here is for defense. So we can have defense against bacteria, which is what you're looking at over in this picture here, defense against a virus, defense against fungi. You can even have defense against cancer cells, as this melanoma here, or defense against something that you might not want defense against, like a skin graft. Really is defense against any cell that has a foreign protein on it, or even something that's not a cell, like a virus. Viruses are not cells, but they do exhibit proteins that are different from your own proteins. And so any foreign protein entering your body or growing from within your body, your immune system is going to try and fight that. So we have a classification here of the immune system. And the first is the innate defense. This is something that you're born with. It's not specific at all. It's just fighting anything that's foreign, not fighting any one specific thing. And there's no memory to it, versus adaptive defense, which is very, very specific. So innate defense is, again, not specific at all. This is just fighting anything that could be foreign. It's not going after any one thing in particular, whereas the adaptive defense is very specific. It is the immune system going after one very specific foreign protein in the body. It's systemic, which means that it's body-wide. Here we're going to be looking at antibodies that travel through the blood plasma body-wide, as well as some T cells, lymphocytes that will also be in the bloodstream, and now, as you know, also in the lymphatic system. And you're going to have some memory to this adaptive defense, where these cells are going to stick around longer so that they can remember in case this very specific foreign protein comes back into the body later. So first, we're going to be looking at the innate defense. The first defense line that you have is your surface barriers. And so in the top right kind of pink box there, your skin is one of the first defenses that you have. It's the surface of your body. It's to prevent bacteria or fungi or viruses from just being able to enter into your tissues. The other surface barrier you have is the membrane that lines the inside of your body, where it's really exposed to the outside world. And this is the mucous membrane. So remember, if you're breathing in air, the air is on the outside environment. Let's say that there's dust or pollen or whatever it is, bacteria, virus, somebody sneezed, now there's a virus in a water droplet, and you breathe that in all through your nasal cavity, all through the oral cavity, all down the respiratory system to your lungs, and then also digestive system. Let's say you eat a Dorito, and that Dorito's outside environment, you eat it, goes down the esophagus. Now that outside environment, that Dorito is in your stomach, makes its way through the intestines until ultimately it comes out through the rectum and anus. So all of that respiratory and digestive system on the inside is really exposed to the outside environment, whether that be a virus in air or if you are eating, what have you. Your mucous membrane is going to be the boundary there. The mucous membrane is the epithelial tissue, exhibiting those lateral cell junctions like desmosomes and tight junctions, and the mucous membrane will also be producing mucus. So these are your surface barriers. We're going to be looking at them in a little bit more detail. Then we'll be looking at the internal defenses that you have inside the body that are present at birth. They are not specific, they don't have memory, but they do help you fight foreign proteins in the body. And then later on, we'll be looking at the adaptive defenses. So here we go, a little bit of a self-quiz for you. What is our first line of defense? I'll give you a minute to think about it. Hopefully you thought of the skin and the mucous membrane. It's your first line of defense to try and prevent a foreign cell or virus from being able to get into your body. Second line of defense are these internal defenses here, these antimicrobial proteins, fever, inflammation. We'll talk about these coming up. And then the third line of defense is, of course, your adaptive defense, where your body is really fighting something very particular, very specific, in order to prevent an infection throughout the body. Here's the first line of defense. Over here you have the cutaneous membrane, known as the skin. Now, one way that the skin tries to prevent bacteria from overgrowing, the skin is going to be about a pH of 5.5. Remember that a pH of 7 is neutral. So 5.5 is slightly acidic, and this is going to help prevent an overgrowth of bacteria on the surface of the skin. Now, when you think about the vagina, about a pH of 4, stomach, about a pH of 2. And so pH is one way that we can really try and prevent bacterial growth, part of our first line of defense there on the skin. The mucous membrane was lining all the internal passageways, so the respiratory system here, down the trachea, into the lungs, as well as the digestive system, down the esophagus, down into the stomach, and continuing on through the intestine. This is going to be a boundary to try and prevent bacteria, viruses, fungi from being able to cross through. Again, a major function here is to produce mucus. So if you breathe in dust particles and it goes into the trachea, now remember there's cilia there. Hopefully that dust particle will land in the mucus lining the trachea, and the cilia beat in an upward motion to move that dust trapped in the mucus now up through the larynx and over to the laryngopharynx and down the esophagus so that it can make its way down to the stomach where it has a pH of 2 to help degrade bacteria, etc. So this is a first line of defense, preventing things from getting across. It's serving as a boundary. Now, when the first line of defense is penetrated, then that's when your second line of defense acts. So here, we're still going to use our innate defenses. We're born with these. They're not fighting any one particular foreign antigen or foreign protein, but just fighting anything that is a non-self cell or anything that exhibits a non-self protein on the surface. Now, when you have tissue damage being done, there are inflammatory chemicals that get released. It's going to make this blood vessel a little bit more leaky, so more fluid can go out into there, which is why you get some swelling, you get more interstitial fluid buildup. Remember that these white blood cells, these happen to be neutrophils, are going to slow down as they travel through this blood vessel under the exposure to this inflammatory chemical. They slow down with those cell adhesion molecules, CAM, C-A-M, cell adhesion molecules. They squeeze out of this now more permeable blood vessel. Now remember, if this is a continuous capillary, normally entire cells cannot get out through a continuous capillary. In this case, we have inflammatory chemicals, which is triggering this capillary to now become more permeable than it is typically. And so we have white blood cells squeezing out. Just as a review, remember that's called diapedesis. Then remember there's positive chemotaxis, moving toward a chemical. The amoeboid movement, of course, this is all review. Remember that these neutrophils are phagocytic. They can engulf that bacteria, merge the vesicle with a lysosome, which contains digestive enzymes. So again, it's not going after any one particular antigen on the surface of that bacteria. Just anything foreign will trigger this phagocytosis to occur. All right, phagocytes, remember site means cell, are part of your internal defense. We're going to go through more of these internal defenses looking at natural killer cells, inflammation, which is going to be a review for you, antimicrobial proteins, and fever. So phagocytosis, again, this is review. You first learned about this in Bio 32. And this image here happens to be a macrophage. Remember, a macrophage is a monocyte that is out in the tissue, so it changes names. Macrophage is phagocytic. Here, it's going to engulf some kind of pathogen, and it wraps the plasma membrane of the macrophage around this until it totally pinches off. And now when that happens, pathogen is inside of the vesicle. Now, this is a phospholipid bilayer here as the boundary of the vesicle. This phospholipid bilayer used to be on the surface of the cell. It used to be here, if you follow the mouse, and it kind of concave, kind of pulled inward and wrapped around and pinched off, and now it's inside the cell. So you take that vesicle and you merge it with a lysosome. Again, there are digestive enzymes in here. It says acid hydrolase. ACE on the end is going to be an enzyme. And then you're going to degrade whatever in there. Then you can spit out the parts at the end through exocytosis, and now you have killed that pathogen that was in the body. So phagocytosis is not specific, but one way to fight an infection of any protein that's not a self protein. And again, that term that we're going to give for that is antigen. It's a non-self antigen. Here's a natural killer cell. This is a type of cytotoxic lymphocyte. But what happens here is that when a cell that doesn't have a self-recognition protein on it is considered foreign, as this embryo is here, What the natural killer cell does is it injects something into the target cell that triggers apoptosis. Now, apoptosis is not a term that you've heard before, at least in Bio 33. Apoptosis is a regulated cell death where the cell kills itself off. And there are some chemicals that can cause a cell to kill itself. And so natural killer cells will inject this into the target cell. And then when that's injected into the cell, the target cell will kill itself off. And again, the term that we give for that is apoptosis. Inflammation is another non-specific immune function. We've looked at this before, where you're going to have an inflammatory chemical that gets released. This makes capillaries more permeable. Things can leak out, including white blood cells can squeeze out through diapedesis. They're going to leak some fluid out into the tissue. We call that exudate. There's some nutrients there to help repair the tissue eventually. But with the extra fluid being leaked out into the interstitial space, you're going to get some swelling, some localized edema. And with that swelling, that can put some pressure on some pain receptors, leading to the sensation of pain being felt. With this increased permeability, another thing that happens is a dilation of arterioles. Now remember, when a blood vessel is severed, the epithelial cells lining the inside of the blood vessel, and we now know that as the tunica intima, those cells are going to release a paracrine, and the paracrine is going to travel not very far, which is why it's not deemed a hormone, but it's going to trigger to the smooth muscle in the tunica media, causing it to constrict very briefly. And so you do have a brief vasoconstriction, but again, it's brief. And then these inflammatory chemicals will actually cause neighboring arterioles to dilate and get larger for the sake of bringing more blood to the area, because with more blood to the area comes more white blood cells and more nutrients to try and repair this tissue. And so when you have that arteriole dilating, we have local hyperemia. So this is an increased blood flow locally to the area. And with that blood, of course, you're going to get some redness due to the color of the hemoglobin, and also some heat. It's going to be carried with the blood. So these are the 4 hallmark signs of the basic inflammatory response: heat, redness, swelling, and pain. Again, this should be reviewed for you, but part of the innate defense. So we had that nail that was stuck in the tissue. An inflammatory chemical gets released. We're going to have some dilation of the arteriole, increased permeability of the arteriole. And so now, at least in the capillaries anyway, you will have some white blood cells leaving the blood supply, going out through diapedesis, moving through the tissue with a amoeboid movement, moving toward those inflammatory chemicals with positive chemotaxis, so that we can fight any bacteria that happen to be in there through phagocytosis. All right, so from your lecture notes, go ahead and look at this page. This is kind of a review about what we were just talking about. So inflammatory mediators here. Again, arterial dilation, that means the inside lumen is getting bigger. So if an arteriole is like this, it's going to get a little bit bigger. Increased blood flow. With that blood flow is going to be more heat, more redness. But now, looking back at more recent things that we've learned, if you have arterial dilation and getting larger, now what you should be thinking is that's going to be a drop in resistance. And a drop in resistance, of course, is going to increase flow, which is why we have more heat and redness. And again, that word hyperemia, localized increased blood flow to the area. And so this is review, but tie in the new information with flow equals pressure over resistance. If you drop resistance, the bottom number, by increasing the diameter of the blood vessel, then you're dividing by a smaller number, which means that your flow is going to increase. And again, that's going to bring extra nutrients to the area. Now over here, I mentioned that capillary becomes more permeable. It's the middle box here. And that really has to do with histamine. Histamine is going to act on the epithelial cells that are making up the endothelium, or the tunica intima, of the capillary. And when histamine acts on those cells, the cells actually start to lose water. And when they lose water, the cells then shrink because they're losing volume. And so they shrink away from their neighboring cell. Remember that the epithelial tissue exhibits cellularity, which means that one cell's up against the neighboring cell, they're touching. And so if the cells shrink due to water loss, then they're actually kind of pulling away from each other as they shrink, which makes them more leaky. The capillary is more leaky. And so we get this fluid from the plasma leaving and going out into the interstitial space, and we call that exudate. By having extra fluid in there, it's putting pressure on the tissue. You're having a localized edema, and that actually works to trap pathogens in that area. By having a tissue swell, now it's harder for pathogens to get into the blood supply because the tissue itself is swelling up. It creates a larger distance now for, let's say, bacteria to travel from the wound site into the nearest blood vessel. But of course, this local edema can cause pain and swelling. And if there's so much pain due to the swelling pushing on the pain receptors, somebody might not want to move because, again, the joint movement might put additional pressure on that pain receptor, making them feel like their limb is immobile. Now, chemotaxis, on your notes here, it says attraction. Really, attraction is going to be what's known as positive chemotaxis. Chemotaxis just means travel, think taxi, travel toward or away from a chemical. And so positive chemotaxis is a attraction toward that chemical. There is also negative chemotaxis. That's not what we're talking about here, but it exists where something's going to move away from a chemical. But we're talking about positive chemotaxis here, and that's going to bring white blood cells to the area, like we saw in the previous photo, like neutrophils, in order to eat up bacteria that's there through phagocytosis. Number 2 on your list there is clean up debris. Really, that's a monocyte leaving the bloodstream, going out into the tissue. Remember that when monocytes go out in the tissue, we rename them macrophages, even though they're the same cell. Macrophages are going to eat up dead and dying cells. So when the neutrophils go out there and they eat up all these bacteria through phagocytosis and they become spent, the neutrophil itself can die, and now you need to clean up those dead and dying cells, and that's what a macrophage will do. You can also have release of more inflammatory chemicals. These are called interleukins. We're going to come back to these later. But anyway, you're going to have fluid, cellular debris, white blood cells that are potentially dead, and they're all going to create what we call pus. And remember, white blood cells are white, and so the pus is white. When we see them under the microscope and they appear purple, remember that's because they're stained purple. They're really white in your body. But all of these are necessary for healing, to get rid of the bacteria and to clean up dead and dying cells so that the tissue can start to repair. So there's also antimicrobial proteins that are part of the innate defense, and this is really cool. So we're going to start with a virus outside of the cell. The virus is going to enter the cell, and it really needs to do this in order to replicate. The virus kind of hijacks the cell's internal mechanisms in order to replicate itself. And then the virus can leave the cell and go into other cells and now continue replicating and just hijack the cells in your body to be virus-producing machines. What's really neat is that when a cell has a virus that gets into it, what can happen is that a gene on the DNA will become activated, and we're going to undergo transcription and translation of this gene that codes for a protein called interferon. When interferon binds, again, a protein, so it's going to be a surface receptor, it will trigger that neighboring cell to start to produce an antiviral protein. So now the neighboring cell is going to produce transcription, translation, and make an antiviral protein to block the virus from replicating inside of it. And so it's really, really neat, this interferon, where one cell says, oh, shit, I'm infected. And then it's going to tell its neighboring cells, it's coming, make some antiviral protection for yourself to try and prevent the virus from replicating in them. So really, really neat. This is called interferon. We can actually inject interferon into people who have hepatitis C to try and make their cells produce antiviral mRNA and antiviral protein to fight against that hepatitis C virus, which is really neat. Another type of antimicrobial protein is a complement, and this is really many proteins working together. This says MAC right here. That stands for membrane attack complex, and it's multiple different protein components that are going to arrange and insert themselves into a cell that they want to destroy. And by embedding this into the membrane, now you can have diffusion in and out of the cell through this big pore that was created. And now the cell can no longer regulate what's in and what's out. And so the cell can then fill up with water and lice and rupture. And so this complement protein, which is all these multiple proteins here that start with C, as well as this MAC, membrane attack complex, can end up lysing cells by jamming this complex into the membrane of the cell that they want to target. So another antimicrobial protein there. Fever is another way that your body has an innate defense to try and prevent the replication of pathogens in the body. So remember that enzymes are very temperature specific. And so enzymes are going to catalyze chemical reactions that would otherwise not happen at a speed fast enough that is necessary. This pathogen that enters the body is going to need a certain temperature for its enzymes to work properly so that it can cause an infection in your body. Well, if you raise your body temperature to be higher, hopefully that means that the enzymes are going to denature, and now that pathogen is not going to be able to survive as long inside of your body, which is a way to decrease the infection. And so let's look at the bottom here. White blood cells detect a foreign invader. They produce something called pyrogen, which is a very fun name. Pyro is somebody who likes fire. So pyrogen is going to trigger your body to increase its set point. So remember, the hypothalamus has a set point for your body temperature, about 98.6. Although in recent research, they've actually found out that people's body temperatures have been slowly lowering over the decade. The average body temperature is not really 98.6, it's actually slightly below that. But at any rate, this pyrogen is going to trigger the hypothalamus to say, you know what, the set point's not at 98.6. Now the set point is at 102 or 103. And so you still have a negative feedback mechanism going on of trying to maintain your body temperature at a set point. It's just that the set point has changed and now it's higher. And so the body maintains its temperature at a higher temperature, which is going to make you feel miserable, but it's going to try and prevent the pathogen from being able to do well in your body. Of course, this works to a certain degree. If you get up to 104, 105, it becomes dangerous for your own body. 106, extremely dangerous. All right, so we've looked at the innate defenses here. Your first line of defense being your surface barriers, your skin and your mucous membrane, of course, both epithelial tissue. And then you have the internal defenses of your phagocytes, like your macrophages and your neutrophils. Your natural killer cells, remember, they inject a chemical into a target cell to trigger apoptosis. That's the term that we give for the target cell killing itself off. Inflammation response, the antimicrobial proteins, we looked at interferon and the MAC complement proteins there, and then fever. Once you have those innate defenses, then we also have something called adaptive defenses, which is going to help your body fight not against any foreign invader, but against a very, very specific protein sequence on the surface of some foreign invader. And your cells are going to mount a specific response against it. It's attacking something that it's been shown, hey, this is a foreign antigen, go and attack this. And then the cells will have very specific proteins that only match that antigen to attack only that antigen on whatever pathogen surface it happens to be on.